Hello, everybody. How are we doing? Are we good? I can see you talking in the chat about results day. Not long to go. What is it, about 10 days? Oh, you're all gonna do fine. Guys, what grade have you got to get to be able to do A-level biology? It's probably different, right, in different schools and colleges. What have you got to get? Is it a grade six? Grade seven? Six for you, Annie. Grade seven, yeah. Seven, seven. Oh, seven, seven for combined. Oh, thanks, Kira. Oh, you're going to write me a good review. Wow, thank you. You have to get nines because you're doing fast track A levels. You just need to get a five. Yeah, different schools tend to have like different entry criteria, don't they? Grade six in maths, yeah, Aria, that's quite common, isn't it? Because there's quite a lot of maths in A-level biology. So a lot of places do put a requirement on the maths as well. I know a lot of schools say you've got to get a six in maths to do A-level biology. You're all going to do fine, I'm sure. You'll have to let me know when we have our lessons next week. You'll have to let me know how you get on. Um, oh, we've got in year 11. Is this going to be relevant? You can see for sure, like a lot of it you'll understand. A lot of what we're doing in this course, let me just explain, especially if any of you are new to the year 12 course. We're doing some like looking back at GCSE stuff and then building on it for A-level biology, right? So if you are only in year 11 or only going into year 11, some of this you'll find hard, but if you're hoping to do A-level biology, then by all means, you can give it a go, right? See how you get on. Thanks, Kira. Um, Recommending to everyone, perfect. That's what we want to hear. We want recommendations. We want to get loads of people on our courses next year so we can help as many people as possible. Yeah, so the last two lessons that you've had with me, we've done like A-level cell stuff, right? We've done like A-level animal cells and plant cells. We're going to build on that today and we're going to focus on specifically the organelles involved in protein synthesis and the transport of proteins in the cell. So some of these you'll know from GCSE, but some of it is just A-level content. Okay. I'm not going to introduce myself again because you will know who I am by now, I'm sure. We're going to get straight into the lesson if that's okay. So, for this slide, what I want you to tell me, I want you to name as many organelles as you can remember that you think are involved in protein synthesis, so making proteins, or transporting proteins. So think about your GCSE organelles, but also think about the stuff we did last week and any A-level organelles that are involved in making proteins or transporting proteins. Okay, so a lot of you are giving me ribosomes, obviously, because ribosomes we knew about at GCSE and we know that they make proteins. Good. Some of you are also saying the rough endoplasmic reticulum, which you won't know from GCSE, but we learnt about it last week. So the rough ER for sure, it has ribosomes on its surface, so it's obviously involved in making proteins. Some of you are saying the Golgi body or the Golgi apparatus, which is correct because the Golgi body is involved in modifying those proteins. So after they've been made, they might want to be modified in some way. Anything else? We've got vesicles. Good. Because vesicles are involved in the transport of those proteins from the ribosome to the Golgi or from the Golgi to the cell membrane. Right, so some of us are saying nucleus, which I think is correct because the nucleus contains DNA, which codes for proteins. So it's definitely involved, isn't it, in making proteins. What about the nucleolus? Can anyone tell me the role of the nucleolus? I'll write it down here. What was the specific role of the nucleolus, guys? Zainab's given me all of them. Nucleus, Ruffia, ribosomes, Golgi apparatus, vesicles, and cell membrane. Good. Nabil, good jazz. The nucleolus makes ribosomes. So you could say it must be involved in making proteins because the nucleolus makes the ribosomes and then the ribosomes makes the proteins. So without the nucleolus, you wouldn't have the ribosomes to make the proteins. What about, oh, cell membrane? Yeah, we could add cell membrane. I think Zainab said cell membrane. We could add that because the cell membrane is involved in the transport of proteins out of the cell. We're going to go through each of these, by the way, so you don't have to write the functions yet. We're just getting our little lists together. What about the mitochondria? I'm going to add that to my list. Does anyone know why the mitochondria are necessary for making proteins? Think about the job of the mitochondria, think about what it releases, and think about why that is vital for manufacturing proteins. Good. Zainab's saying ATP. Any other answers? ATP is what releases energy, right? Energy needed for synthesis. Good. Releases energy. And energy is needed to synthesize anything. Yeah? Because to make proteins, you're going to be making bonds that is going to require energy. Okay, powerhouse of the cell. Yeah, we all say powerhouse of the cell, but don't ever say it in an exam, Toby, because it's not going to get you marks. The other thing just to remember is we shouldn't say produce energy, because producing energy we cannot do. We cannot make or destroy energy. So we have to say release energy instead, okay? But yeah, release energy, which is needed to make proteins. So at GCSE, we really only talk about ribosomes being used to make proteins. But what I'm now telling you is all of these organelles are in some way, shape or form, involved in the manufacture of proteins, yeah, and the transport of those proteins. So we're going to go through this now in a lot more detail, and we're going to think about each organelle. Good. Now, when I was looking through this presentation this morning, I noticed a mistake on this image. There is one organelle on this image that should not be on this image. Can anyone tell me which one of these organelles is not involved in protein synthesis? Because we're going to cross it out. Exactly. You've all spotted it. The smoothie R. I think I got a little bit carried away with all these organelles. The smoothie R is actually not involved in protein synthesis. Can anyone remember the job of the smoothie R? Because we'll write it down just as a reminder, but I'm going to cross it off this particular image. Okay. What was the job of the smoothie R? Exactly. It makes lipids. A few of you given me the correct answer there. It makes lipids. It makes fats. So we're going to cross it off. It's not directly involved in protein synthesis or the transport of proteins. Okay, let's cross it off. So the ones we're going to talk about then today, and I'm just going to kind of use this image to circle them again. We're going to make notes in a sec, but we're going to talk about the role of the nucleus in protein synthesis, the role of the Ruffia, and obviously the ribosomes. We could draw some ribosomes on, couldn't we? Should we draw some onto our Ruffia? Remember, the Ruffia is called rough because it has ribosomes attached to its surface. So we could draw some on there. And there are also some ribosomes free in the cytoplasm. Okay, so there's two types of ribosome, isn't there? Free in the cytoplasm and on the Ruffia. So we're going to talk about those. We're going to talk about vesicles. We're going to talk about the Golgi body. And we're going to talk about the cell membrane, or Do you know at A level, guys, we should really call it the cell surface membrane, if we can remember to, because there are lots of cell membranes. The nucleus has a membrane, the rough ER has a membrane, the Golgi body has a membrane. So we should really specify when we mean the cell surface membrane, we should really put that in there. Okay. Is it hot where you are, Aria? It's actually quite cold where I am today. It looks pretty grey and rainy. Okay, so we're going to go through each one of these in turn. Let's have a look at the questions. A, is the rough ER, the vesicle and the Golgi body in the cytoplasm free or attached to something? So all of these organelles are in the cytoplasm. The rough ER, the ribosomes, the vesicles, the Golgi body, they're all in the cytoplasm. They are kind of held in place. I wasn't going to talk about this today, but seeing as you've asked the question, A, the organelles in the cytoplasm, don't know whether you know this yet, but they are held in place. Does anyone know what by? We've not mentioned it yet, so I don't know whether you will, but all of these organelles that are in the cytoplasm are held in place. Does anyone know what by? Kind of fibres, yeah. Does anyone know the name of those fibres? Good, the cytoskeleton, which is made of protein fibres. So they are kind of held in place. That's the role of the cytoskeleton, or one of the roles is to hold these organelles in place. Another question, what's the difference between the plasma membrane and the surface membrane? Nothing. So you can call at A level, if you prefer to call the cell surface membrane the plasma membrane, that's absolutely fine. Okay, you can call it the cell surface membrane or the plasma membrane. Perfect. Okay, so let's go through these organelles and see if we can get the functions. Remember, we're focusing specifically now on its role in protein synthesis, okay? So there's loads of things you could tell me about the nucleus. In fact, we spent about 15 minutes in our first lesson talking about the nucleus. But if I'm going to say to you specifically, what is the nucleus role in protein synthesis? What are you going to say to me? And I'm going to cross off this one again because I think I've repeated the same diagram with that incorrect smoothie R on there. So what is the role of the nucleus just in protein synthesis? We're not going to talk about the nuclear envelope or the nuclear pores really. We're just going to focus on the DNA. Good Zainab, good CS, good Nabil, good Aria, good Havana. Right, okay. So let's write this down. So the first thing we're going to say is the nucleus contains DNA. Now, a lot of you are telling me that the DNA codes for proteins. Can anyone remember specifically which bit of the DNA codes for proteins? Which bit of the DNA? Think about the structure of DNA. And yes, Delight, you are right. Transcription happens in the nucleus, so we can put that down in a second. Okay, MZA, yes, the bases. 3 bases codes for 1 amino acid. Yeah, a lot of you, yeah, genes. A gene, CS, a gene is the code for a protein, but within that gene, it's the bases that determine the order of amino acids. Okay, so we're going to try and be that specific. So the nucleus contains DNA. DNA contains bases. And it's the bases that determine, I'm trying to be as concise as I can here, the bases that determine the order of amino acids in a protein. So I'm trying to be really specific. What is the role of the nucleus when it comes to making a protein? Okay, the nucleus contains DNA. DNA contains bases, and you're all right, by the way. 3 bases, you've got some good memories. 3 bases code for 1 amino acid. So we sometimes call it a triplet or a codon. Dylan, well done. A triplet or a codon, like a set of 3 bases. but it's the bases that determine the order of amino acids in a protein. Okay, a tricodon, not a tricodon, it's either a triplet or a codon. A triplet or a codon. Someone also said for the nucleus, we could make a comment that the nucleus contains a nucleolus, which makes the ribosomes. And obviously ribosomes are needed to make proteins. So you could give that as another function of the nucleus. Okay, you were taught tricodons. I have never heard that before. I've never heard tricodons before. So tRNAs. So tRNAs, they actually carry the amino acids to the ribosomes. If you remember that from GCSE, you might do. tRNAs carry amino acids to the ribosomes. So they're not in the nucleus. Okay, can you remind me of chromatin? Yeah, for sure. So chromatin was a term we mentioned last lesson. Chromatin is the collective name for DNA and histone proteins in the nucleus. So the nucleus, we would say, contains chromatin. And then you could go on to say chromatin is made from DNA. And DNA contains bases, okay? But chromatin is like the collective name for the DNA wrapped around proteins, okay? Does that make sense? Aliyah, I think, asked me that question. Okay, but yeah, this is my simple summary. If I was writing an exam answer on the role of the nucleus in protein synthesis, I'd probably focus on the DNA, the bases, and the fact that the order of bases is ultimately going to determine the order of amino acids in that protein, okay? And then extra little mark, maybe the nucleolus, which is, look, the pink bit here. Shall I label the nucleolus? The nucleolus also makes ribosomes, which are needed for protein synthesis, okay? Any more questions about the nucleus, or are we happy to move on here? I think I'm managing to keep up with the chat quite well today. You're all being very sensible. Histine. Oh, that says histone. Is that my writing? These are called histone proteins that the DNA is wrapped around. What is mRNA, please? Good question. Yeah, a histone protein is what the DNA wraps itself around. So mRNA is made in the nucleus, okay? Now, when we learn about transcription and translation, we'll do that in a lot more detail. But basically, the mRNA is like a copy of the DNA. So you've got your DNA base sequence, and then the mRNA is a copy of that DNA base sequence, messenger RNA, yeah. And that's what leaves the nucleus and goes to the ribosomes. But that's a little bit more in depth than what we're doing today. That's when we focus on transcription and translation, which is a little bit later on in year 12. Okay, that's like module 4 of year 12. Okay, whereas this is basic cell stuff, which is module 2. Okay, so you'll probably get onto this in like October, November time, okay? Right, so that's the nucleus. So that's where we're starting. Let's move on to the ruffia and the ribosomes. Tell me what you would say, yeah? If you were doing an exam and you were asked what was the role of these organelles, remember, just in making proteins, what are we going to say? And these two answers are going to be very similar because the ruffia has ribosomes on its surface, okay? So they link together. I'm just going to make a little note. The ruffia has ribosomes attached to surface. Okay, so the ruffia manufactures, nice word, or synthesizes proteins. And so do the ribosomes. The reason I've split it up is because we've got two types of ribosomes. We've got ribosomes like these ones that are in the cytoplasm. And we've also got, if I do some little red dots, we've also got ribosomes on the rough ER. So both of these organelles actually make the proteins. Okay, Delight, I'm going to add what you've said there because Delight has added something extra on for the rough ER. The rough ER not only manufactures and synthesizes proteins, but it also transports proteins to the Golgi body. Does anyone know the difference, by the way? I'm just going to pose this question to you. Does anyone know the difference between the proteins that are made by the ribosomes in the cytoplasm and the proteins that are made by the ribosomes on the rough ER? Does anyone know why we have those two types or what the difference might be? The diagram might help you. The ones that are made on the rough ER ribosomes are going to go to the Golgi, and then they're going to go to the cell membrane. Whereas the ones made by the ribosomes in the cytoplasm Good, Aaliyah. I hope I'm saying your name right, Aaliyah. Ribosomes made in the cytoplasm stay there. Oh, sorry. Proteins made at the ribosomes in the cytoplasm, I assume you mean, stay there. Yeah. This is kind of beyond what you need to know, but I think you're kind of understanding me. So let's continue with this idea. Proteins made by ribosomes in cytoplasm stay in the cell, and proteins made by the rough ER get modified by the Golgi body and usually leave the cell. Yeah, we're going to do the Golgi body in a second, so don't worry, because you weren't here for the last lessons, were you? That's the difference. So if it's a simple protein that doesn't need to be modified and is just going to stay in the cell, like maybe it's an enzyme that is used inside the cell. If it doesn't need to go anywhere, it can be made at the ribosomes in the cytoplasm, okay? Because it's just going to stay in the cell. It's not going to be modified. If it needs to be modified, so if it's quite a complex protein and it needs to be modified, or maybe it needs to be, I don't know, made into a glycoprotein, so maybe it needs some carbohydrate added to it, it's going to have to be made by the rough ER, because then it can go to the Golgi body and be modified. And then the Golgi body can send it to the cell membrane and it can leave the cell. Okay, so this is for proteins that need to be modified and often need to leave the cell and go somewhere else. Okay, thanks for clearing up that. Yeah, I was wondering if any of you were kind of thinking, why do we need ribosomes in the cytoplasm and ribosomes on the rough ER? Just depends on the protein. Does it need to be modified? Does it need to leave the cell? Because if so, it's going to be the rough ER that makes it. Okay. Yeah, so ones that become part of the cytoskeleton might be made by the ribosomes in the cytoplasm because they're going to stay inside that cell. But yeah, a lot of proteins need to leave the cell. They need to be secreted. Like imagine if it's a hormone, guys, it needs to leave the cell and go into the bloodstream. So it needs to go via the Golgi body and through the cell surface membrane. Wait, but the proteins are made in the ribosomes on the rough ER. That makes the protein not the actual protein. I don't understand that question. Can you type that in again? What type of protein leaves the cell? A hormone would be a really good example of a protein that leaves the cell. An antigen would be another one because an antigen has to be put outside the cell on the cell surface membrane. Some enzymes have to leave the cell. Like enzymes made by the pancreas have to be secreted out of the pancreas into the small intestine. Okay. The GCSE class today is at 1 o'clock, isn't it? 1 till 2. But the proteins are made on the ribosomes, not the actual roughy-R. That makes the I still don't understand your question, eh? Try type it out again from scratch. Okay. Let's move on to the next organelle. So we've got the nucleus. We know the nucleus has the DNA which codes for the protein. We know the ribosomes and the roughy-R then manufacture the proteins. Then if it's made by the roughy-R, remember the roughy-R transports proteins to the Golgi body. And then what is the role of the Golgi body, guys? Okay, A, I get your question. Are proteins made in the roughy-R as well as on the ribosomes that are on? No. The roughy-R only makes proteins on the ribosomes that are on the roughy-R. Okay, so the roughy-R has ribosomes on it, and it's those ribosomes that make proteins. Okay, perfect answers. So the Golgi body can modify proteins. That's one thing we can say. They can package proteins. I'm going to put into vesicles. And then transport proteins in the vesicles. Okay, so modify, package, secrete, yeah, modify proteins, modify, transport, good. Processes, yeah. You could also say modify proteins, package proteins. They also allow you to say process proteins. Now, a few of you have mentioned lipids as well. You are correct. They also modify lipids and package lipids and process lipids. I'm just not writing that today because this lesson is focused on proteins. But you are correct. The Golgi body can also do this with lipids. Okay. You thought the roughy-R packages and transport proteins. So does the Golgi body do the same thing? Very similar, Dylan. So the roughy-R makes the proteins and then transports them to the Golgi body. The Golgi body can then modify the proteins, but then, yeah, it does package and transport them as well. So they're both involved in the transport of proteins. They're both involved in the packaging and transport of proteins. But the difference is the roughy-R is making proteins and then packaging and transporting, whereas the Golgi body is modifying proteins and then packaging and transporting. Okay. Does every protein go through this step? No, not every protein, because if it's a very simple protein and it's just made at the ribosomes in the cytoplasm, then it's not going to be modified and transported out of the cell. Okay. Yeah, so CFK is talking about the cytoskeleton. Yeah, the cytoskeleton is a network of fibers. It does give strength to the cell. Yeah, it holds the organelles in place as well. And it does allow the movement and transport. Yeah, all of these vesicles, by the way. So, I've got this blooming smoothie R on there again. All of these vesicles. So once the rough endoplasmic reticulum has made the protein, packages it into a vesicle, which goes to the Golgi body. The Golgi body then modifies the protein, packages it into a vesicle, which goes to the cell surface membrane. All of these vesicles, as CFK has just said, are moved using the cytoskeleton. Okay, so they're moved along these fibers. Good, Dylan. Yes, that would be a really good description of the Golgi body. It modifies proteins and lipids. It transports them to vesicles or packages them into vesicles, and then it transports them. Okay, what is the Golgi body? So it's an organelle, which you don't learn about at GCSE, unfortunately. It's made from flattened membranes. It almost looks like sacs, flattened membranes, but its role is what's important, okay? So its role, we're going to say it modifies proteins and lipids, if you want to just put that down. It packages proteins and lipids into vesicles, and then it transports proteins and lipids in those vesicles. Is it packed in micelles? We're talking about lipids now, Sarah. No, micelles are something that's formed during digestion. So when we digest lipids, we use bile, and the bile salts and the lipids is what we call a micelle. So it's not quite relevant here. Does radiation damage the DNA? Yeah, it can do, can't it? It can cause mutation. Yeah, and then that can ultimately affect the protein that gets produced, because if you mutate the DNA, you're going to change the order of bases. That can change the order of amino acids in the protein. Good question. Right, so the secretory vesicles then transport the proteins. And again, if you want me to remind you, I can say, and the lipids, to the cell surface membrane. By the way, I have put a little question on this PowerPoint later on where we are going to put all this together. So we're going to write it out as a nice story. I love a biology story. I love it when we can just learn like a whole story. It's nice, okay? So the secretory vesicles, obviously, once the Golgi body has modified the protein, packages the protein, puts it into a vesicle, those vesicles can then transport that protein to the cell surface membrane, okay? Does it stay in the vesicle after leaving the cell? No. I'll do you a little picture. I think, have I got it on the next slide? Exocytosis. It is through exocytosis, Sarah and Andreas. Yes, it is. So it doesn't stay in the vesicle. The vesicle kind of fuses with the cell membrane and releases the protein on the other side. And that is called exocytosis. Which organelle makes lipids? No, the smooth ER. We're not really focusing on lipids today, but the smooth ER makes lipids. The rough ER makes proteins. And then the Golgi body modifies the lipids and proteins and packages the lipids and proteins. Is translation involved here too? That would be at the ribosomes, right? So when we make the proteins at the ribosomes or on the rough ER, that's translation. That's basically when the amino acids actually get joined together. That is protein synthesis. Okay. Trying to cover a lot here. We will put it all together, so don't stress if you're still thinking, I'm a little bit confused and I want it all written down nicely. Okay, we are going to do that, so just bear with me. Oh, in fact, have I done it for you? I think I've done it for you and we're going to do it ourselves. Okay, I'm very kind. Right. I'm going to annotate a little bit though, right? Because what I've done here is I've put a summary together, but I have done it really simply to give us the opportunity to add a little bit more. So the nucleus contains DNA, but we need to remember, because this is A level, DNA contains bases. And the bases code for sequence of amino acids. Okay, that's A level standard. We can't really just say the DNA codes for protein, okay? We should know that it's the bases in the DNA that determine the order of amino acids in the protein, okay? Let me know if you've got any questions about these steps. Then the ribosomes synthesize the proteins, make the proteins. If someone just asked me a second ago, this stage would be called translation, making the actual protein. So that would be on the rough ER or the ribosomes in the cytoplasm. Okay. Now the rough ER is then going to package proteins into vesicles, and then they go to the Golgi body. The Golgi body can modify the proteins. We can say sort the proteins as well. E.g., I've given you one example of a modification. Some proteins have a sugar chain attached to them. A glycoprotein is basically protein plus carbohydrate chain. Now that's an example of a modification done by the Golgi body. So it takes the protein and it sticks on a carbohydrate chain, a glycoprotein. Then the Golgi body puts the proteins into secretory vesicles that are usually transported out of the cell. They might stay in the cell, but they're usually transported out of the cell if it's a hormone or an enzyme. The vesicles often move to the cell membrane, and then the proteins leave by exocytosis. Okay, so the way this works is the vesicle fuses with the cell surface membrane and releases the protein. If you've not heard of exocytosis before, that's what the contents is going to be secreted. That's a really good question. So the protein inside that vesicle, might be a hormone, for example, it's going to be secreted. Secretory vesicle. Can you hear me now? What's exocytosis? Yeah, you can hear me. Exocytosis is basically when something is released from the cell. Yeah, exo. Exo means, think of where you've heard exo before. Where have you heard exo before? Oh, I've got pins and needles in my leg. I think I've been sat on my legs this whole lesson so far. I've got pins and needles. Yeah, exothermic. So doesn't exothermic means to like release heat energy? So exocytosis means to release something from the cell. So yes, Andreas, good question. There's also endocytosis. Endo is where we're taking something into the cell. So endocytosis is going in, and exocytosis is releasing out of the cell. Yeah, so that makes sense, doesn't it? And yeah, I've also added a bit of extra knowledge that vesicles do move through the cytoplasm using the cytoskeleton. Exoskeleton, like insects have an exoskeleton. It's on the outside, it's outer, an outer skeleton. Are the proteins made by the ribosomes in the cytoplasm packaged into vesicles? It really depends, Ange, or is it Ange or Ang? It depends what those proteins are. If they do need to be transported to another part of the cell, then they might be packaged into vesicles. If they are going to be used in the cytoplasm, then they are made and released straight into the cytoplasm. So they don't need to be packaged into vesicles. So the protein going into a vesicle. No, I'll show you what endocytosis. So here's my cell. So say there's something in a vesicle outside of the cell. So let's draw something in this vesicle. If the vesicle fuses with the cell, like this, I can't do this drawing without rubbing the whole thing out. If the vesicle fuses with the cell membrane and brings the substance into the cell, that would be endocytosis. So the vesicle comes from outside, maybe from another cell, and the vesicle fuses with the cell membrane and the substance is carried into the cell. That would be endocytosis. whereas exocytosis is the opposite. Okay. Okay. What time are we on? Right, we're going to do some questions and we are going to get a chance to see what we can remember from that whole story, okay? So I've given you some kind of straightforward-ish. They are A-level questions though, don't get me wrong. These are not GCSE, these are A-level exam questions, all of them, okay? Giving you some more straightforward ones and then we're going to do like a 5-marker, okay? Because at the end of paper 1 for A-level, you get at least 3 5-mark questions, 5 or 6 markers, and this topic comes up quite a lot there, okay? So, can we place the letters in the correct sequence to show the passage of an amino acid from the cytoplasm at F until it is secreted from the cell at the cell surface membrane, which was K. So here's F, which is obviously the cytoplasm. And we've got an amino acid there, just a single amino acid. We want it to get all the way to K, which is the cell surface membrane. So we basically want to get all the way to exocytosis. Why are there 6 letters but only 5 in the box? Because you don't need to use all of them. Some of them are there to trick you, right? Some of these letters, like G maybe, is there to trick you. So we've got an amino acid. Where's it going to go first? Then to where? Then to where? Before it gets to the cell membrane. Okay, I think you've got this right, guys. H, J, E. E are not ribosomes. These little things here are ribosomes. E is a bit too big to be a ribosome, right? Said it first, but missed out the H. Right, let's just go through and name the organelles though. So F is just the cytoplasm, and it told us in the question we had an amino acid in the cytoplasm. Now that amino acid is going to get joined into a protein. So what organelle is H, guys? What is H? Good, Aliyah, yeah. Aliyah. I'm not sure how to pronounce it. Yes, Zainab. So H is the RER. So here, the amino acids are joined to make proteins, okay? Because you can see there are ribosomes on the surface of the RER. You can see the dots on the surface. Then we're going to J. What is J? It's gone from the rough ER to J. Good, Dylan. The Golgi body. Now, the Golgi body might modify the protein. It's going to package the protein for transport. Then you've gone to E. What is E? Obviously, K is the cell surface membrane. So what was E? Well done, vesicles, or Dylan even said secretory vesicle because it is being secreted out of the cell. So we can call it a secretory vesicle. That vesicle is basically going to transport that protein to the cell membrane so it can be released through exocytosis. So at K, the protein is released through exocytosis. Okay, so we've gone rough ER to Golgi body to secretory vesicle to cell surface membrane. Okay. Yeah, good work. Your GCSE, you're giving it a go. Good on you. Yeah, the mitochondria, I don't know whether you can see down there. The mitochondria was labeled down in the bottom corner. Now that does release energy, and that energy is needed for protein synthesis, but the amino acid is not going to go to the mitochondria, right? It's not directly involved, is it? It's not going to go to the mitochondria, but the energy is needed from the mitochondria. Okay. Let's have a look at another question. So this cell is found in the pancreas, apparently. Which type of protein will this cell manufacture? Now, this is worth 2 marks. So I would like the type of protein and a named example. And there are two options you could go for. And in fact, some of you have already given me both of those options. Are they only called secretory vesicles if they are going out of the cell? Usually, Natalie, although you don't usually get penalised for it. Yes, secretory vesicles are usually carrying it to the cell surface membrane. But to be honest, you can just call them vesicles or secretory vesicles. They don't really mind. So, well done. Which types of protein? So we've got hormones and enzymes. Now, can I have some named examples, please? Well done. So for hormones, you should be able to name insulin and glucagon, because this is the pancreas, right? It makes hormones to regulate your blood sugar. What enzymes could you name? Amylase would be correct. Protease would be correct. Lipase jazz would be correct. Well done. So it's making a lot of proteins, this cell. Well done. Not thyroxine, because thyroxine is made by the thyroid gland, which is a little gland in your neck. That's the one that controls your metabolic rate, right? Which you might know from GCSE. So thyroxine is made by your thyroid gland, not the pancreas. Okay, so bearing that in mind, we're saying, look, this cell makes loads of proteins. It makes hormones, it makes enzymes. Look, it's going to have loads of rough ER. It's going to have loads of ribosomes. But why does it also contain a lot of organelle G? What is glucagon? So glucagon is a hormone that your pancreas releases when your blood sugar is too low. If you've been exercising or fasting, your pancreas would release glucagon. Okay. So, organelle G. So, organelle G is a mitochondrion. Now, you're not going to get a mark just for saying that, because it's not asked you to name it, but you should recognize it, right? So, what are we going to get marks for saying? Mitochondria release energy through what type of respiration? What type? Oh, good CFK. Good. Release energy through aerobic respiration. Now, because this is an A-level lesson, if you prefer, you could say mitochondria makes ATP through aerobic respiration. But don't worry if you've not said that yet, because we haven't really learned about ATP yet. But aerobic respiration makes ATP, and then the ATP releases the energy. That's how it works. ATP stands for adenosine triphosphate, because it has 3 phosphate groups. So at A level, we start to learn a little bit more about respiration. And instead of just saying respiration releases energy, we say respiration makes ATP, and then ATP is actually what releases the energy. So obviously this cell needs lots of mitochondria because they release energy through aerobic respiration. And then we need to say, for 2 marks, protein synthesis, or making proteins, requires energy, or we could say ATP. Okay, any reaction where you are making bonds, and here we are joining amino acids together, we are making bonds between amino acids to make a protein. I'm sure you know from chemistry, if you're making bonds, it uses energy, it requires energy. I get confused between adenine and adenosine. Easily done, easily done. So adenine is a base in DNA, isn't it? And adenosine is in ATP. But did you know that adenosine does contain adenine? Kind of going off on a tangent here, but seeing as you're asking such good questions, adenosine is basically ribose sugar and adenine together. So that's why it's confusing. because adenine is just a base in DNA, but adenosine is a sugar and that base. Okay, it's ribose sugar, which is a 5-carbon sugar, joined to an adenine base, and it's called adenosine. Yeah. Okay. Right. I think we've covered everything needed for that one. So let's just finish off with a couple more questions. For question 1, is an amylase produced more in the salivary glands? Yeah, amylase is produced in the salivary glands, but it's also produced by the pancreas. The pancreas produces a few different carbohydrates, enzymes, but yeah, it is definitely produced in high quantities by the salivary glands. Yeah, maltase is another carbohydrate. Isn't adenosine what is used to stop your heart? Not that I know of. I don't know what you're thinking about there. Not sure what you're thinking about there. Okay, we're going to do a 5 marker. Now, I'm not going to expect you to give me like a huge paragraph in the chat. So what I'm going to suggest that you do is choose it's 5 marks. So we're telling a story. We're telling the whole story of the production and the release of enzymes, which are proteins. So we're basically telling the whole story of the making and the release of proteins from a cell. You can smash this question. Pick an organelle. I mean, if you're feeling really confident, you could do it in order. You could go nucleus, ribosomes or ruffia, Golgi body, vesicles, cell surface membrane. You could do it all if you want, but I'm going to suggest you pick at least one organelle and give me the specific function in terms of the production and the release of proteins. This was an A-level exam question, okay? It's on A-level paper 1. At the back of A-level paper 1, the last 3 questions are always this style, okay? They're always like 5 or 6 markers. They're nice. They are biology stories. That's what I like to call them. So start putting some notes in the chat. I'm just gonna write down the organelles that you could get credit for talking about. Because there's a lot of organelles you could get credit for. I'm going to try and do them in order. You can also get credit for mitochondria, by the way. Look, I'm not taking my own advice here, am I? I'm meant to say cell surface membrane. Okay, so these are usually the ones on the mark scheme for this kind of question. Now, obviously, if you just list the organelles, you probably get 1 mark just for listing organelles. They want you to describe the role. Okay. Is anyone going to give me anything? I'm waiting. I want you to do it. Just take one of them if you want. Just take one of them and tell me the specific role. Okay, good. Jawar here has started us off. Should we say enzyme or protein? We could say either, couldn't we? The bases determine the sequence of, I'm going to say enzyme, just so the examiner knows that I've paid attention to the question. I'm going to say the bases determine the sequence of amino acids in the enzyme. You could just say protein, don't get me wrong, but I'm trying to say to the examiner, I've read the question, I know we're talking specifically about an enzyme. Nice. Okay, keep going, guys. These are good. CS, the amino acids. So translation does not occur in the nucleus. Translation is when we actually join the amino acids together. So that would be at the ribosomes. Okay. So you'd say that here. You could say the ribosomes or the ruffia synthesize the proteins, or you could say that that's where the process of translation occurs. Because translation is where the amino acids actually get joined together. Okay, yes, and you could also say transport of proteins to Golgi. I'm running out of space. I hope you can figure out what I'm... Okay, so the ribosomes, the ruffia, synthesize the proteins, or it's where the process of translation occurs, where the amino acids get joined together. And for the ruffia, you could also say it's going to transport the proteins to the Golgi body. So the Golgi body, we're going to say modifies. There's so many words we could use. You could say modifies, sorts, processes the proteins, and packages the proteins into vesicles. Okay, yeah. Then the vesicles, what we're going to say, transport the proteins. I'm only talking about proteins because the question is about an enzyme, okay? So I'm not going to mention lipids at all. So transports the proteins or enzyme to cell surface membrane, okay? Now, the reason I've got mitochondria here is we could say releases energy or makes ATP, which is needed for protein synthesis. So even though the amino acids don't go there and the protein never goes there, we still need the mitochondria to release that energy. And then the cell surface membrane releases the enzyme via exocytosis. Have I got everything there? That's basically what you learn. You learn it off by heart, and there is a lot of memory involved in A-level biology because you have to recall some of these complex stories, okay? But this question was specifically about enzymes, so we're just focused on proteins, and it does say that they are being released from the animal cell. So we do need to get all the way to the cell surface membrane and exocytosis. Okay, vesicles move to the cell surface membrane where it leaves by exocytosis. Perfect, Jawar here. Because the question is directed to enzyme production, would it be wrong to say the ribosomes in the cytoplasm? No, because some enzymes do stay in the cell. So you could just mention ribosomes in the cytoplasm. However, because this question specifically wants you to get to the release of the enzyme, can you see that? Release the enzyme from the animal cell. You definitely need to also talk about the ruffia, the Golgi body, the vesicles, and the cell surface membrane. Okay? Because it does have to leave the cell. You could still mention ribosomes. They're not going to penalize you if you say, oh yeah, and there are ribosomes in the cytoplasm that make enzymes. They're not going to penalize you, but make sure that you go down the whole ruffia, Golgi body, cell membrane route. Okay? Have we had a chance to get these notes down? Remember, guys, you cannot always watch the video again, rewind the video after the lesson or whatever. But this is what you need to learn. This is what, if I had like a lesson objective or a learning objective, this is what I wanted you to learn. I wanted you to be able to tell me exactly which organelles are involved with making proteins and what they do. Okay. Oh, we've got some more questions. Should we do them super quick? Have we got time, guys? I think these are quite easy, right? Describe how the ruffia is involved in the production of enzymes. These are easier than the one we've just done. So enzymes are proteins. So I think that's all they're testing you on. They're checking, do you know that an enzyme is a protein, right? So the Ruffia synthesizes proteins like enzymes. These are proper A-level questions, by the way. I've not made them easy on purpose. Okay. So it synthesizes proteins because enzymes are proteins, and it transports them to the Golgi body. Describe how the Golgi apparatus is involved. Is this... Okay, I'm just going to say something about this question. If you just told the examiner everything that the Golgi body did, that's not really what they want, okay? Yes, the Golgi body modifies proteins. Yes, it sorts proteins. But it specifically asks how the Golgi apparatus is involved in the secretion. So it only wants you to talk about what the Golgi body does that helps with the secretion of proteins. Does that make sense? So you can say it modifies them, you can say it sorts them, but that's not going to get you the marks. What do you think they want you to focus on? How the Golgi body is involved in the secretion of enzymes. Good, Nabil. Perfect. And again, you could say proteins or you could say enzymes. I'm going to say for transport to cell surface membrane. Okay, because we're focusing on secretion, right? So it's going to package them into a vesicle, which will be transported to the cell membrane, cell surface membrane, to be secreted. Okay, nice. Yeah, okay. For the 5 marker, could we write that mRNA is made in the nucleus? You could, transcription. Yeah, you could say mRNA is made in the nucleus, but I think you would still have to get across the point that that mRNA codes for the amino acids in the protein. Okay, so yes, mRNA is made from the DNA. That's the process of transcription, but the mRNA contains bases that code for amino acids in the protein. Okay. Yeah, you can ask some more questions. Right, if you want to go, that's absolutely fine. You can go. We have finished. I hope it's been useful. Make sure you do your homework. I'm going to say it every lesson. It's only a super quick homework, but make sure you do it. And then enjoy the rest of your lessons. Right, what are your questions? Someone had some questions. See you on Wednesday. See you on Wednesday for lesson 4. Yeah, if you've got any questions, just put them in the chat. I can stay for a couple of minutes. That's fine. Thanks, guys. Thank you. I hope it's been useful. What is the rough? So RER stands for rough endoplasmic reticulum. And it's an organelle that isn't on the GCSE spec. But basically what you need to know about the rough ER is it's a series of membranes with ribosomes attached. Okay, so it can make proteins because it has ribosomes stuck to its surface. So those ribosomes make proteins and then they get transported through the series of membranes and then they go to the Golgi body. Okay. Do you have to mention the nucleolus for the 5 marker? No, you didn't have to, because the nucleolus makes ribosomes, right? But the question was about making enzymes specifically. So yes, you do need ribosomes to make enzymes. And yes, the nucleolus does make those ribosomes, but you didn't need to mention the nucleolus for a 5 marker. How does it make the protein? So just like ribosomes do, it joins amino acids together in a process called translation. So tRNAs carries, these are called transfer RNAs. So they carry amino acids to the ribosomes which are on the endoplasmic reticulum, on the rough endoplasmic reticulum. So transfer RNAs carry amino acids to the ribosomes, and the amino acids are joined together. That process is called translation. Translation. So one amino acid gets joined to the next. There are bonds. The bonds between the amino acids are called peptide bonds, which is something that you'll also learn in year 12. And next question, when you say they are packaged enzymes into vesicles, what exactly? So if you imagine you've made your enzyme, so here's my enzyme, which is a complex protein with an active site. Basically, the Golgi body, which is also made of membranes. So the Golgi body is made of membranes. Some of this membrane kind of buds off and wraps itself around the protein. So a vesicle is basically just a membrane bound sac. So some of the Golgi body membrane kind of buds off with the protein. So the protein is like enclosed in a membrane, and then that vesicle can carry it to the cell membrane. Does that make sense? Honestly, if you're year 11, don't be stressing yourself out, but hopefully if you're going to go on to do year 12 and A-level biology, this will be helpful. What are so a vesicle is dead simple. It's a membrane. It's literally just a membrane. It's made out of lipids. It's made out of phospholipids. It's like a little sphere of phospholipids, just enclosing the protein. Okay. Right, I'm going to have to go because I've got GCSE at 1. But yeah, have a good one. I will see you guys on Wednesday. Thank you so much for all your good questions as well. Bye, guys.